All right, in this video, we are going to talk about the experiment that you'll be doing in lab regarding invertase. And so the big picture here is that you're going to be characterizing a protein. What protein are you interested in? Like we just said, invertase. And when you are studying a protein, there's all sorts of questions that you can ask. You might want to know what's its amino acid sequence. You might want to know what is its structure. Might want to know where it is located in a cell and what is its function in a cell. So you've learned about invertase from the primary research article assignment that you did last week. And you know that invertase is an enzyme that catalyzes the hydrolysis of sucrose into glucose and fructose. Or remember, enzymes catalyze both directions of a reaction. It just depends upon what the delta G is. And so it is quite possible for invertase to catalyze the formation of sucrose by glucose and fructose. Now, in today's lab, it is very important that you understand the direction of the reaction that we're going to be looking at in the context of the organism that we're going to be looking at is in this forward direction, is invertase catalyzing, speeding up the reaction in which sucrose is broken down into glucose and fructose. So we know a lot about invertase already. We know it's an enzyme. We know its structure. So what question are we trying to answer today? Well, we are trying to answer the question of where does invertase function within yeast, Saccharomyces cerevisiae. This is a eukaryotic organism. It is single-celled. It's the same yeast that you have here for baking. It's the same yeast that's necessary for making beer and bread and things like that. Now, here is the two alternative hypotheses that we're going to be working with here. And the scientific question is invertase intracellular, meaning does invertase work inside the cell and there it breaks down sucrose into glucose and fructose? Or is invertase extracellular? Is it a secreted enzyme, meaning does the cell make invertase and then secrete it into the extracellular solution where it breaks down sucrose there in the extracellular solution? So we have the intracellular hypothesis and we have the extracellular hypothesis. One thing to note, and this is going to be important for all of the logic of your experiment, in that if our intracellular hypothesis is correct, if invertase stays within the cell and breaks down sucrose there in the cell, then glucose and fructose are going to accumulate in the cell. If, however, invertase is secreted, if yeast secretes invertase into the surrounding solution, and there it breaks down sucrose, then our glucose and fructose is going to accumulate in our extracellular solution. Okay? Right, so now, how are we going to find invertase? What is our assay? And our assay, the way in which we are going to find invertase, because here's the thing, folks, that the ultimate way to find invertase, find out where it's functioning, is a direct assay where we are using a high-powered microscope and looking at the cell and go, oh yeah, look at that, the invertase is here within the cell. Or, oh, look at that, invertase has been secreted into the surrounding environment. We simply don't have that technology, certainly not at OCC. So we need to take advantage of some other fact about invertase in order to track where it is at any given time. So let's remind ourselves that invertase takes sucrose and breaks it down into glucose and fructose within yeast cells. So let's take a look at the chemistry of glucose and fructose. And what you'll notice is both of them are considered reducing sugars. And what that means is this carbonyl carbon here in glucose, this carbonyl carbon here in fructose, it has the ability to donate electrons to other stuff. That's what it means to be a reducing sugar. However, sucrose is not a reducing sugar. Aha! So what invertase does then is we can sort of reterm this, is what invertase does is it takes a non-reducing sugar, sucrose, and it converts it into two reducing sugars, glucose and fructose. Okay, we're getting one step closer. Now, all we need is some sort of test that can distinguish between reducing sugars and non-reducing sugars. And that is called the Benedicts reagent. Okay, so what the Benedicts reagent does is it is cupric ions. And when those cupric ions in the solution here are boiled with, are heated in the presence of reducing sugars, two things happen. First, you get this beautiful color change, this sort of red or orange color change, very specific. And second, you get this precipitate. So you can kind of see through this liquid here, right? It's colored, but it's still translucent. Here, what we have is precipitate that is clouding up the solution. So a positive Benedict's test is one in which the color changes to sort of an orange or red color, and you get precipitate forming. And that tells us then that we have the presence of reducing sugars. Sucrose will not test positive for Benedicts. All right, so we're on to something here. We're on to something. We now have a way of determining where invertase is located, because wherever it's located, it's going to turn sucrose into glucose and fructose. And if it does that, then we have reducing sugars when we didn't before, and then we're going to have a positive Benedict's test. Okay, so that's the indirect way we are tracking where invertase is. We're tracking the accumulation of its products, and if we see them, they're reducing sugars, they'll give us a positive Benedict's test, then we know invertase must have been there. So now the question is this. In order to figure out, what we want to do is we want to separate the intracellular fraction, that's going to be the cells, from the extracellular fraction. That's going to be the liquid here, because we can't just add Benedict's solution to this whole thing. That's not going to tell us anything. Right? We need to first separate the yeast from the surrounding solution. The yeast cells from the surrounding solution. The intracellular fraction from the extracellular fraction. How do we do that? The way in which you separate dense things like cells from solution is with a centrifuge. Now what a centrifuge does is it's got this rotor in here, and you're going to put your samples into the centrifuge, and you're going to close that up, and then you spin it, right? This rotor spins very, very quickly. And what that does is it forces all the sort of the more dense particles to the bottom of the tube, and the less dense liquid stays towards the top there. And so after you spin, and you pull your sample out, what you're going to see is, look at that. All that at the bottom, those are all the cells that were initially equally dispersed within that solution, but because we centrifuged it, they all went down to the bottom. That's called the pellet. So no matter what you centrifuge, when you pull it out, that bottom solid portion is called the pellet. That's going to represent our cells. That's going to represent the intracellular fraction. The top part, that's called the supernatant. That's going to represent our extracellular solution. It's going to represent what the yeast cells were incubated in. Okay? So now what we'll be able to do is you take that supernatant and you pour it into another tube, right? You get to separate it out. And that leaves behind the cells, like that. And so what that will allow us to do is then add Benedict's solution to the cells only, add Benedict's solution to the extracellular fraction, so basically add it to the pellet, add Benedict's to the supernatant, and boil it, okay, heat it up, and see which one of the two fractions, the intracellular fraction, the pellet, or the extracellular fraction, the supernatant, which one of us gives a positive Benedict's solution? And let's make some predictions here. If the extracellular solution gives us a positive, right? A positive Benedict's test means the presence of glucose and fructose, means the presence of invertase. So, if we find it in the extracellular, if we find it in the supernatant, that means invertase must have been secreted into the extracellular solution and there broken down glucose, sucrose to glucose and fructose. If, however, our pellet is positive for the Benedict solution, if our cells test positive for the Benedict solution, that means that glucose and fructose accumulated within the cells. That means invertase was within the cells, and that means that's where it broke down sucrose. And so that means invertase is intracellular. This is the logic that you are going to be using to figure out, to answer our scientific question: is invertase intracellular or extracellular?